so my responsibility is focused ion beam, but I'm working now with Nikki also on cryotransmission electron microscopy. So today I want to try to give you a flavor about what you can do with focused ion beam microscopes. Give you a quick overview about the fundamental processes, maybe a little bit of a history. It's a relatively new technique compared to others that you might be familiar with, but it's a constantly evolving technique, and there's a lot happening at the moment. It is a very versatile technique, so you can use it across a broad range of materials, right? So from physics to biology, and I'll hopefully show you a few examples of those. And then hopefully at the end of the talk, you might have a sense for, okay, what are the dimensions that I can work on, right? Is this potentially useful for my application? So at the core of the technique is the interaction of a bundled ion beam, an electron beam that is an ion beam that is focused onto the sample down to nanoscale resolution, and the kinetic energy of these ions makes the ions penetrate into the material, and they induce sputtering. So we have here, it produces, and because we are focusing the beam, we can do this on the nanoscale. So with that implantation, so we are having implantation of gallium, sputtering of the material, and in the end result, we're using it to sculpt the material, to cut the material down to the nanoscale. With those different properties, we have core applications that are used in our facility. The implantation is not so much used in our case. We are really using it for the cutting, right? So there are three main categories that you can divide the work up. We can simply use the ion beam to machine on the nanoscale. We are using it heavily within the facility to prepare samples. It is a helper tool for other instruments like transmission electron microscopy and atom probe particularly. And the instrument has now evolved to be an outright analysis tool on its own. And in particular, it is very powerful in doing 3D imaging on the nanoscale. And I will show you an example later. So as I said, the technique is relatively new compared, say, to transmission electron microscopy or X-ray techniques. It was developed by the semiconductor industry that had the need to look underneath the surface of their structures that they have developed. And the first microscopes were basic single gallium ion columns, right? So basically like an SEM, but you're replacing the SEM column with a gallium column. And then you have the first major component that was built into these tools was a gas injection tool, which allowed the engineers to deposit material, conductive, non-conductive samples, and they have also edge enhancing materials, and these allowed them to edit their circuits on specific locations in their chips. If you're curious about more details, there are at the moment 2 major books out there. They're a little bit outdated, so probably it's time to get a new book out. But the technique has then evolved, and it became very soon clear that a single gallium column alone was rather impractical because you can only cut, but you don't see what you're doing while you're cutting. So the next logical consequence is to combine a focus ion beam with an SEM column, and this is basically we have the dual beam focused ion beam Furthermore, we have now the development of manipulators that allow to extract material. And so this was then basically the onset of preparation of TEM samples. And the evolution of the technique is ongoing. So the standard iron species that we typically use at this moment is gallium. It has the perfect properties to generate the needed fine probe of ion beams, but there are developments in designing new sources that have different properties. For example, simply by using different species of ions. So with those, we now extend, for example, the volume of the material that you can access. For example, a plasma ion microscope has a higher ion current. With the higher ion current, we can mill a larger volume of material with the beam in the same time frame as compared to what we can do with our standard gallium sources. The manipulators started out first ex situ, so we had to prepare the sample and then we used external manipulators to plug these small pieces. The manipulator moved inside, so we can now in situ in the microscope prepare a sample and plug it and transfer and do whatever we want to do. And this was, for example, a crucial development to make a technique called atom probe tomography that my colleague Austin Akey is responsible for and can help me with, to make that really accessible to a broader range of materials. Before, the focused ion beam atom probe was very much limited to metallic samples because they could be drawn into tiny shapes that were needed for the technique. And with the focused ion beam, one could suddenly prepare the material for all sorts of materials. Also, the software has evolved more and more. We became more and more sophisticated. And so nowadays we can do three-dimensional imaging with that technique. We can run the instrument over days and record a lot of data and get three-dimensional information about the material. And right now, the latest developments that are happening right now is that, as you can see, we're adding cryo stages to further expand the range of materials. And as you can see at the bottom, the range of applications is broadening and broadening from starting out with semiconductors, then we got into material science, physics, and now we're also working with biologists. So we do have two instruments at CNS. The workhorse is the Helios NanoLab 660 from FEI or thermal. So here you see it's based on an SEM. So the electron column is the major part of the instrument. And then on the side, on the left side, you see the ion column stick in. These two beams cross over at one particular point, the crossover point. And we do have a stage that allows to place the sample very precisely at that crossover point. As I mentioned before, we do have gas injection systems that allow us to deposit material in situ in the microscope, and you will see in a moment what we're using that for. This is the manipulator that I talked about, and we have a plasma cleaner that allows us to keep the system clean, and we have a whole range of analysis detectors, including EDS for chemistry, EBSD for crystallographic analysis. So overall, it's an extremely powerful tool where we can analyze the sample and sculpt the sample with the ion beam. Together with the Helios, we also have from Zeiss, the Crossbeam 550. And with this second tool, the second tool allows us to expand our range of materials that we can work on. So for example, a major difference between the two SEM columns is that we have here an SEM column with an electrostatic lens at the bottom, which allows us to work on magnetic material, which is not possible in the Helios. But the major difference in terms of applications is that we do have a cryo stage for that tool. And at the moment, this is the only focus ion beam where we can do air-free transfer of material. And so this has become a more and more important topic in the last couple of years. And so we are building right now all the necessary workflows so that we can work on such air-sensitive materials, for example, right? And a key example are lithium batteries, right? So because lithium, if you work on lithium, you not only need to keep it air-free to omit oxidation, but the material is so sensitive that the gallium beam is actually damaging the material and modifying the structure. So with lithium, we also have to work at cryogenic temperatures to produce the sample with the result with the necessary quality. And so we are right now, we have some of these tools already running, and we're building now all the consequent workflows that we need to access those materials. So just to give you a sense of what you could do in terms of nanomachining, so we have here groups like the Kim group at Harvard that works on 2D materials, and they are using the focus ion beam to generate a mask for deposition of electrodes so that they don't have to use clean room methods, so that this is carbon-free, solvent-free process. And this allows them to, so they're cutting the structure, as you can see, into a silicon nitride membrane, and then they can place that electrode arrangement as they need it on those two-dimensional flakes that they have. So they can not only locally place it, but they can also play with orientation. And so they have, this gives them a lot of flexibility to really locally work on very specific 2D material patches. So when you work with the focus ion beam, there are certain considerations that you have to take just think about. So the degree with which we can focus the ion beams depends very strongly on the current, how many ions we apply into the beam. So in the best scenario, at the low current, we can bundle the ion beam down to 10 nanometer resolution. So we can get a structure of down to 20 nanometers that we could form. Then on the other end, when we're on the high end, we are somewhat limited in the total volume that is practically accessible, because we can at max pull out a current of 100 nanoampere. And so based on the sputter rate for the gallium ions, the interaction rate with the material, that in practical sense leaves us to a volume that is on the order of several tens of micron that we can work on in a practical manner. Practical means in a reasonable time frame. And we also need to be aware, right? So if you want to do periodic structures, that is challenged because we are working with a serial scanning process. And then a very important factor, this is sort of often a limiting factor of the technique. As I said before, we are simultaneously implanting gallium and sputtering. So whenever you work with the ion beam, you are modifying the surface that has seen the ion beam. So we are changing the electronic structure, we are generating defects. And so for many applications, you always, whenever you're considering to use a focused ion beam, you need to ask yourself, is this damage or implantation affecting the experiments that I want to do? Yeah. So if you want to do electric measurements, you need to be considered, is that changing the electric conductivity? Is it changing my optical properties? So there are a lot of considerations. These are fundamental considerations that you have to make. Yeah, and so we had applications that we had to stop, right? So there was the idea to generate micro lenses around NV centers, and that had to be given up because the implantation of the gallium generated so many optical states that would have overwhelmed the signature of the NV centers themselves. And I will later hopefully give you a little bit better sense of what that implantation looks like. So at CNS, focused ion beam has become a central tool that is used to prepare material for other instruments, right? And the two major instruments we talk about here are, of course, the transmission electron microscopes and the atom probe. And in particular, preparing TEM samples, that is here in our facility the key application, right? So just to give you a sense how the process works. So as I said, we can deposit material. We can deposit material both using the electron beam and the ion beam. The electron beam has the nice effect that it's not damaging your material, so you can preserve your material down to the last monolayer of whatever you have. So you can actually cover 2D material and prepare cross sections, and you can see in the cross section the actual 2D material like graphene. Yeah. So then we typically put on top of so we are basically generating a buffer layer using the electron beam, and then we add more material on top. So with this we are sort of protecting the region of interest. Then we start with the ion beam to cut trenches in the front and in the end, and we then trim these where we have a rectangle standing in the middle that we then trim down to a certain thickness. Then we are using the ion beam and cut a J-shaped form into the structure, so that we have basically then a cantilever left over that is just barely hanging on a small bridge. So this is now where we come in with the manipulator. We glue the manipulator on the free end with the material that we're depositing, and we cut the bridge, and then we can pull out that piece with the manipulator. Then we transfer the sample onto a special holder that fits into the transmission electron microscope. Glue it on, cut off the bridge. Yeah, so now we have this cantilever hanging like a flag, for example, on one of those arms. And we can then continue thinning it down to the necessary thickness on the order of 100 nanometer of thin. And then in the end, we can see TM images. Incidentally, we do have a scanning transmission electron microscope in the Helios itself. And so you can see here a comparison of the TM image that we recorded with the Helios and the one with our atomic resolution microscope. And you can see on the nanoscale, it produces similar results. Of course, with the Helios, we cannot go down to atomic level, right? But we can technically use the STEM to get a first quality assessment of the sample before going into the actual TM. So I would like to direct your attention to this dark gray area, which shows this kind of model structure. And it actually turns out that what you see there has nothing to do with the sample itself, but what you see there is actually the effect from the gallium that has been implanted into the surface layer of the TEM. So this is here again, this is basically we're going back to the topic that I was talked about at the beginning, right? That we have to be really careful about the gallium implantation and its effects. So what basically happens is that the gallium ions transfer their kinetic energy to the host in a series of scattering events. Eventually, it has given up all its energy and will be implanted at a certain depth. The gallium ions can remove host atoms from their original crystallographic location, and we generate defects. Yeah? And these can then reduce the crystallographic order. We can generate stress, and of course, with defects, we're changing the electronic state. And so ultimately, to give you a sense of dimensions, so if you look here on the rightmost side, the range, that is sort of the medium implantation depth of these ions in the material, right? So we are talking here about order of tens of nanometers implantation. So whenever you prepare a TEM sample, right, you need to be aware that when your TEM sample is, say, 100 nanometer thick, underneath both surfaces, you have a defect zone on the order of 10, 20, 30 nanometers. Right? So that means you have then, let's say, when your defect zone is 30 nanometer, then you have suddenly only 40 nanometer pristine material left over. And so this is here illustrated here with this experiment where I just cut a little trench into a sample. So you see here a little trough, and then prepare the TEM sample of it. And then you can see here in detail, here near the corner, the actual structure. So the mottled structure on the top right is infiltrated material that I produced with the gas injection system. And then this dark band that goes around the corner, that is the defect zone. So this is where basically the gallium ions have, ultimately the dark region represents a zone where the crystallinity has been completely destroyed. This is silicon, and silicon tends to amorphize. So the dark band represents the location where the material has been amorphized. And the brighter regions represent where the gallium is, where the gallium has been implanted. And so there are some geometric effects where there are some geometric differences between glancing incidence and under vertical incidence. So we are a little, so under glancing incidence, the effect is a little less strong. So we can roughly divide the defect zone by a factor of 2 when we're thinking about TEM sample preparation. But nevertheless, these damage zones are quite significant, as you can see here on this comparison, where we have a 5 kilovolt gallium beam, the corresponding defect zone. So on the right side here, where this little, so we can see the crystalline structure on the right side of the image, and then the defect zone in the gallium implantation on the left. And you can see how with the reducing energy of the gallium ions, the defect zone gets smaller. Yeah, because the gallium ions introduce less energy and they are stopped with fewer scattering events. So that is ultimately the strategy, how we are improving the quality of TEM samples. So we typically operate the instrument at the highest energy at 30 kilo electron volt, which produces the sharpest beam and has also the highest sputter yield. But then when we prepare sample, at the end, we go down with the energy and carefully shave off damaged surface layer. But ultimately, even at 1 kilovolt, which is sort of the practical limit on the focused ion beam, we still have quite a bit of defects in the surface. And so ultimately, what we are then using in the end is a low energy ion milling tool where we can polish the sample very carefully with argon ions that have only an energy below 1 kilovolt, so they're typically 500, 700 volts. And there we are now entering a true polishing regime. We can very carefully remove the last parts of the damage zone. And just to go back to illustrate the difference, so this was the image that I showed you before, which was basically how it looks after a typical 5 kilowatt cleanup in the gallium, with the gallium in the focused ion beam. And this is the actual structure as it actually looks like. And you can see now you can see beautiful tiny precipitates in the material that were completely obscured by the mottled defect structure that the gallium implantation has generated on the left side. Yeah. So this is sort of the art of preparing good TM samples. And so here I think we have all the necessary tools to prepare whatever sample you need with very high quality. So as a side note, of course, when the most basic application for focused ion beam, instead of going through that complex process of cutting out and extracting a temp sample, we can just cut a cross section and look at a cross section. In that case, in many materials, we don't need to worry about defects too much. But as soon as we're dealing with soft material that has feature sizes on the order of the defect dimensions, right, on the order of tens of nanometer, the electron signal stems from the defect zone. With that, we can certainly change the morphology. So we are imaging the defective region underneath the surface. And so we have to be very careful. Again, think about, is our material prone to potential damage? So this is, again, soft materials with very small feature sizes. Like in this case, this was an organic-inorganic hybrid perovskite system. We knew from crystallography, from X-ray diffraction, that these tiny nanoparticles that you see here on the left side were crystalline. And our initial imaging was just producing a homogeneous band with no structure inside, because the gallium interaction completely obliterated those little crystallites. So we had to apply the same method of going down with our energy, very carefully polishing that surface, and then suddenly we can bring out those little crystals. Yeah, so this is a key factor that you always have to consider when you prepare with a focused ion beam. So one reason why focused ion beam microscope have become so popular when we are looking at samples with the transmission electron microscope is that we can prepare samples from very specific regions. Yeah, we can be very size specific, and we can now pick samples with much better idea about the overall context. Yeah, so this here is an example from work that we're doing with the Art Museum, the Harvard Art Museum, with Lucy Cooper and Catherine Ehriman. They have vases from the Song Dynasty, and they were interested in the particular properties of these beautiful glazes that these vases showed. And so we did some sort of reverse engineering, if you want. So we got a few chips from those, and they had very particular colors, right? They were reddish and blue, and so they were interested in what actually produced those colors. So we were able to prepare 10 samples from these specific regions and use all the whole tool arsenal that we have available in the transmission electron microscope to analyze the material. And so we found out then that basically there are actually in the red part of this glaze, there are tiny nanoparticles, which are actually producing the red color. And then we could also identify sort of a two-phase material which affects the optical properties of the material and produces a certain effect on the color. So we were able to connect the optical properties of the glaze with the internal nanoscale structure of the material itself. So we can push that site specificness to the limit if we want. So we can again use our gas injection systems as a tool to mark specific regions. And then we can use those fiducial markers to really very specifically go to very fine details. And we can go to very specific regions down to tens of nanometer precision if necessary. Yeah, so it is can be also a very precise tool in that sense. One major development in the past, say, 10 years is that focused ion beam enables us to do 3D imaging on a wide variety, on a large range of dimensions, right? So we have two external techniques. So it helps to prepare samples for atom probe and TM, where we can both allow us to get three-dimensional information on nanoscale and hundreds of nanometer resolution. And the technique itself, via serial sectioning, allows us to get corresponding three-dimensional information up to dimensions on the order of tens of micrometers, but with a resolution of a few nanometers. And it turns out that this kind of volume range is interesting for many applications. So we use it for battery work, for fuel cells in the material science side. So there are quite a few applications on that side. But it has become also more and more important for biological applications. So with biologists are interested in the structure of their cells. And due to the natural complexity of cells, two-dimensional cross-sections often don't reveal the complete picture of the entire system. And this is where the power of serial section focused ion beam comes in, because we can, at least within this small volume, we can get the entire three-dimensional structure. And so this here is an example of work that we've done with the Children's Hospital at the medical school. So the drawing lab works on malaria parasites, and we were able to image the malaria parasites as they have infected blood cells. And so in this particular case, the parasite goes through a very complex transformation as it forms daughter cells. So it turns out that these malaria parasites do not segment in a classical binary way, where one cell generates two daughter cells and so on, but it simultaneously forms 30 daughter cells at one step. So it does that mainly by guiding the outer membrane of the cell, and it will be guided inwards towards the cell. So you can see here in the mid-segmentation image, you can see how there are membranes showing towards the center of the cell. And then eventually these membranes will pinch together. And in the last stage, you have then 30 daughter cells, which then break out of the blood cell and further infect the bloodstream of the patient. And so the research group was very much interested in the details of this evolution. And so we were able to image that with 3D imaging. And of course, they have done a lot of optical imaging, right? So optical imaging is the key tool for biologists. The challenge with these optical techniques is that you can visualize the location of specific components, but often it is then hard to then understand the entire cellular context. And this is where focused ion beam or electron microscopy is still an important tool to augment the information that they get from their optical experiments. And so here's an example. These are 2 volumes. As you then get at the end, once you have taken the series of hundreds or thousands of images, and then you segment these images into the different compartments that the cell has. And then you see here, for example, we can nicely see the early stage. You can see here the outer membrane, how it starts to work inwards. And then you can see certain organelles, how they are distributed in 3 dimensions within the cell. And this type of information you would not be able to get with a simple 2-dimensional cut. Right? So here you have the entire 3-dimensional context of the entire cell. And so this here is now a corresponding volume at a later stage in the development, where you can already see now all the daughter cells have been evolved. The different organelles have been nicely distributed within the daughter cells, and the whole evolution has finished, and this parasite is ready then to break free out of the blood cell. So the next developments that we are working on right now is to make it possible to work under cryogenic conditions, so that we can access materials that typically cannot be analyzed within a vacuum of an electron microscope. Yeah, and so any water-containing material, volatile materials, yeah, sensitive material, right, like lithium that is damaged by the gallium beam. And so this is, for example, an example where we can do a serial section example on a stoma of a leaf. And so this cryofib will be sort of the next step of techniques that we plan to develop and make accessible to the users. So with that, I'm at the end of the talk. I hope I could give you a sense of how you can use these focused ion beams to machine your material, prepare samples, to extract samples, what the characteristic dimensions are, and how you can use it for different applications. And with this, I'm happy to answer any questions. So there. So there was a question in the Zoom. So how thick were the sections in the parasite example? So these were 20 nanometer thick sections, and the images were recorded with a 5 nanometer resolution. So this seemed to be a nice compromise that we do. If necessary, if we need high resolution, we can also make the slices, say, as thin as 5 nanometer, with the constraint that we need to make the volume smaller. Are there any questions?
Would you be able to, you think, section, say, a whole culture cell, something that's 10 microns thick, surreal, or is that beyond?
Yeah, so 10 micron is a 10 micron large cells are realistic. Yeah, so we can go up to, so it's ballpark, a volume of 30 micrometer cube is what we can access with, say, a 5 nanometer resolution. And this is these are parameters that you can play with. Yeah, so you can, if necessary, you can make the voxels a little bigger, and then correspondingly you can access larger volumes. Right, so there are a few parameters that you can play with. So the dwell time, how long you stay on one pixel, the pixel step size, and so in the end you always have to optimize those parameters for your specific application. You know, in some applications you really need the very high resolution. You really need to push the limits down to 5 nanometer, which is sort of the limit for this technique. And so in that so when you need this high resolution in order to do this experiment in a stable way, you need to shrink the volume correspondingly. So these factors all work together.
But when you say 5 nanometers, is that how much has been milled off?
Yeah, so we can mill. So when you prepare cross section phase, yeah, there is enough precision to mill just 5 nanometer away.
And how deep into the sample then is the image from?
So that this is also ballpark 20-30 micron is the practical limit.
No. But how far does the imaging beam penetrate into?
Oh, this is also on that order. Yeah. So we're using backscatter electrons to bring out the staining contrast, and the interaction volume for those electrons is also on that order. So that's where everything fits together.
You know if there's been any efforts to extract like implanted gallium, like for the NV, for the lenses, for the NV centers? Because gallium makes all these cool liquid metals, right? That the gallium typically deep using any chemistry, or do you know if anyone has tried to?
I think they tried to do that. Yeah. So they tried to etch away the top layer. But then in the end, as far as I can tell, from what I know, the whole overhead that came with it made it impractical. Because you then have to do line edge and all these things. Yeah. So there's a question about from Deborah about paper samples. So yeah, technically, so focused ion beam is surprisingly applicable to a wide range of materials. So technically, paper might be, can be also looked at. So we also have, so it always boils down then to the dimensions that you need. So we do have, also as an alternative to focused ion beam, right, where the limit is 30 micron, the volume that we can look at. We do have a cross-section polisher. And with a cross-section polisher, you can look at least two-dimensional cross-sections in paper. But we should be able to, I think there are examples of paper for focused ion beam.
Do you know if focused ion beam microscopy has been used to image or screen other diseases and structures?
Sorry?
So has there been research or studies using this microscopy to image other diseases similar to malaria or just body structures?
Yeah, so you can basically any type of biological material, as long as you can prepare it with the focused ion. So what I haven't mentioned is how we prepare the sample, right? How the samples are prepared, and so fortunately, for the focused ion beam, we can apply the same techniques that are used for transmission electron microscopy, that biologists have used for decades. So basically, when you have your organic material, you first dehydrate your biological material, you replace the water with alcohol, then you replace the alcohol with plastic, and in that process, you add staining material with heavy metals, and those stains go to very specific locations in the cell. Particular membranes are attractive spots for those stains, and so that's the main process, right? So you have then ultimately your material in the end embedded in an epoxy, and in this state we can work on it. And so it doesn't matter what biological material it is, right? So so of course there are then details, right, that I haven't gone too much into in this overview, where when you have tissue material, right, or the challenge with this classical sample preparation is that, as you can imagine, this is an elaborate chemical process, and often the structure has changed a little bit from the original aqueous state. And so one always, if you use that type of technique, sample preparation, you have to be careful and be aware that it can affect the structure, and that you might introduce artifacts. That what you think is part of the structure is actually an artifact of the sample preparation. But there is enough knowledge nowadays, right, where in the community to understand actually the key factors. But there is now, with the advent of cryofilm, a completely new way is now opening up how we can prepare samples, how we can image samples. So nowadays, with cryo, it's possible to vitrify the original organic tissue with its aqueous stage. Vitrifying means we are cooling it so quickly that the water freezes in its amorphous state. With this freezing process omits crystallization. It's so fast that there are no crystals forming, which are not destroying the biological material. So you can preserve the original biological material very close to its original state. And it's possible to actually image the natural contrast between the organic material and the ice. So we can now image with the cryofit, we can image samples in the most closest to the natural state. Right? And so with these techniques, we have now access to a large variety of biological materials. Right? So I'm working right now with colleagues from Mass Eye and Ear who work on hair cells in a mouse or in human hair cells. So there's a whole variety of samples that can now be analyzed. And that's the reason why focused finding has become more and more popular among biologists. That's that?
Yeah.
You mentioned like the technology like using gas injection to add in circuits. Like I just want to know like what's the minimum size of a chip design like we're editing by using this technique like this.
So it's pretty much the same dimensions like I told you about 3D imaging.
Mm-hmm.
Yeah, so the typical working volume that is practically accessible is tens of microns. Yeah, with a gallium fit. So that is a limitation for many applications, right? For many applications, you might be more interested in hundreds of microns or even to the millimeter range. And so this was one of the motivations to develop new ion sources. And so nowadays, many facilities are now using a so-called plasma fit. So where the ion source are conductive ion plasma sources, where we can use nitrogen, oxygen, and xenon is sort of what nowadays is the equivalent to the gallium. These sources allow you to produce a much higher current, and with the current, the sputter yield is scaling up. And so suddenly, instead of tens of microns, you can mill with those instruments hundreds of microns in the same time frame.
Like will the semiconductor material matters like where the size?
This is really surprising how should I say? Yes, the interaction between the gallium or whatever ion you have, there are differences between different materials. Yeah, so definitely when you are working in in a chip, right, the metals react differently than the oxide, than the semiconductor. But this is actually a second order problem. So in first order, it's surprisingly similar behavior. So the cutting behavior in itself is the same, right? It is just then the sputter yield is changing, for example, right? So very hard covalently bonded or ionically bonded materials like diamond, sapphire, strontium titanate, they are tough. So they have a very low sputter yield. But then on the other hand, materials with a very low sublimation energy. So that's basically a nice factor that you can look at, right? How much energy do you need to remove an atom from the surface? So then there's the other side of the spectrum, like gold, for example, melts very quick, right? And so these lead then to more practical considerations. Yeah, so when you prepare cross-section planes, which are very inhomogeneous, with materials with very different milling rates, then you suddenly get into the practical problem that the surface that you're polishing is not nice and smooth, but the soft material will recede first, and the harder material, it's tougher to get rid of, and so you start to generate surface topography, and then that makes sometimes the imaging difficult. Yeah, so on first order, it's quite remarkable that you can actually apply this method to such a wide variety of materials. But then when it comes to working on the material, there's always a little bit of a learning curve, and you need to get a sense of how your material is behaving, and then you just adjust your process a little bit to that. Questions? If not, then thanks so much. Goodbye.